Hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Ma'am, and in today's video we will be completing cell cycle and cell division in less than 20 minutes. So from this particular chapter, 4 questions are expected. So 4 into 4 is equals to 16, and sometimes it can be more than that. And this particular chapter is also required to understand the gametogenesis and some topics of genetics. So let's get started. So when we talk about the cell cycle, it's a sequence of events. It is in a proper order. It is in a proper sequence in which cell has to duplicate its genome, that is its genetic information. So here we consider the haploid content of that genetic information as well, because it will also have the same genes. Then the synthesis of other constituent of cell, and eventually the cell will divide into 2 daughter cells. So when you talk about the cell cycle, right, its duration can vary from organism to organism. For an example, in yeast it is for 90 minutes, and in the case of human it is for 24 hours. So when you talk about the cell cycle, here you have the I phase, which is also known as interphase, resting phase. It is also known as invisible phase. It is also known as preparatory phase, right? And here M phase is the actual division phase. So cell will spend more than 95% of its lifespan in the interphase, because here it is preparing itself for the division, because in the division there should be no error. That is why. So here the quick points are, as I said, cell is preparing for the division, will undergo both cell growth and DNA replication in an orderly manner. So basically in the interphase, the cell growth will also be there, and the replication will also be there in a proper order. And as I said, more than 95% of the lifespan of cell will be there in the interphase, and it is the actual division phase, where the first thing is karyokinesis, which is also known as nuclear division, and the cytokinesis. So karyokinesis, for that we have the trick here, that is PMAT, prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase, that's what we have to remember here. Now let's see this particular diagram and let's understand the things in relation to that. So again, as I said, in the interphase, it is the phase in which what is going to happen, my dear people? It is the phase in which cell growth and the cell duplication will be there. So in the interphase, we discuss it like this. We start with the G1 phase, then we talk about the S phase, then we talk about the G2 phase. That's what we have. G1 phase, S phase, G2 phase. G1 phase, S phase, G2 phase. So G1 phase is also known as gap 1 phase. It is also known as growth phase as well. So it is a phase in between mitosis and initiation of DNA replication. That is a PYQ. Okay. So just to simplify it more for all of you, you people can write it. There is a line in the NCERT that I phase, that is interphase, is a phase present in between 2 successive M phase, right? It's a phase that is present in between 2 successive, that is one after another, M phase. So in the I phase, you have G1 phase, you have S, you have G2 phase. So this G1 phase, because it is after division, after mitosis, so it is also known as post-mitotic phase, and this one is also known as pre-mitotic phase. And actually it is the S phase, where the DNA will duplicate its genome and the number of chromosomes will remain same, and the centriole duplication will also occur here. So this is as per NCERT. So let's have a quick revision. So G1 is the phase in between mitosis and initiation of DNA replication. Cell is metabolically active. Cytoplasmic growth is there. All the enzymes required for replication will be synthesized there. The ATP, nucleoside, whatever is required will be synthesized here, and most of the cell organelles will duplicate. So here in this particular phase, my dear students, right, the duration of this particular phase is variable. It depends upon the cell need. So S phase, as I said, DNA duplication, but chromosome number will remain same, and centriole, which is a non-membrane bound organelle which is present in the animal cell, will also get duplicated here. Then after that, G2 phase is there. Again, the cell growth will continue. Protein formation will be there. So if I have to specify, the tubulin synthesis will occur here only. Okay. And in the S phase, histone and non-histone proteins, mainly the histone proteins, they get synthesized here in the S phase. So this is for the G1, S and G2 phase. So as I said, the chromosome number will remain same here, but the DNA content will get doubled. So if the initial amount of DNA is 2C, it will become 4C. But if initially the chromosome number is 10, it will stay 10 only. It's just that your chromosome, which was earlier like this, right, single arm monad, it will become di-8. So it means the DNA content will get duplicated, but the number of chromosome will remain same. So it's not mandatory that always the cell will divide. It's not at all mandatory. Sometimes there is no signal for the cell division, so cell will exit the cell cycle. Actually in the G1 phase, here there is a restriction point or the start point, right? So if a cell has crossed this, definitely that will enter in the S phase. Otherwise it will leave it. It will enter in the G0 phase. That is basically the cell arrest phase, cell quiescent stage it is. So cell is metabolically active. It's just that it will not divide, it will not proliferate. So it is inactive in sense of proliferation, that it will not replicate, it will not form two cells. But yeah, when it comes to the metabolism, it will be there. So we have the example of heart cell, we have the example of nerve cell as well. Now, after that, right, after this G2 phase, okay, there are checkpoints which will make sure that everything is going fine. Then M phase, which will firstly have the karyokinesis. So when we talk about the karyokinesis, you know it very well. The trick is PMAT, that is prophase, metaphase, anaphase, and telophase. So firstly, let's talk about the mitosis, then let's target the meiosis here. So when it is the mitosis, you know that it is the equational division. Why do we call it as the equational division? Because the daughter cells will have same number of chromosomes just like parent cell. If parent cell is having 10 chromosomes, daughter cell will also have 10 chromosomes. So that's why it is equational, because daughter cells will have same number of chromosomes just like parent cell. That's what you have to keep in your mind. This is what mitosis is. This is what equational division is. So it can be there in the diploid cell, it can be there in the haploid cell. Done. So let's start with the prophase. So whenever you read about the prophase, just do not forget one thing, that whenever it is the prophase, there is the condensation of chromatin material, be it the prophase of mitosis, be it the prophase of meiosis 1 or meiosis 2, the condensation of chromatin material is something very important. Centriole will start moving to the opposite pole. So centriole non-membrane bound organelle, they will be there in the animal cell only, and two centrioles, when they are perpendicular to each other, plus when they are covered with pericentriolar material, they form centrosome. What do they form? They form centrosome. It is also known as cell center or cell sphere or diplosome, that will form spindle apparatus. So chromatid, both the chromatid of chromosomes are visible in this particular stage, right? The microtubules will also assemble, because centriole is also moving to the opposite pole. The microtubules will also assemble because they have to form the spindle apparatus. So at the end, Golgi bodies, endoplasmic reticulum, nucleolus, nuclear membrane, they all will disappear. Then moving to the metaphase. So basically when it is the prophase, in the prophase, just keep it in your mind, the nuclear membrane is disintegrated. All the chromosomes are there in the cytoplasm. Yes, they are attached to the microtubules because spindle apparatus has formed. They are attached to the microtubules. But the one thing here is, that is something you have to remember. Whenever you talk about the chromosomes, you know that this is the centromere. So from both the sides, it is covered with the disc-shaped kinetochore, and to which, right, that's a PYQ. So this shaped kinetochore is there, to which the microtubules are attached, the spindle fibers are attached. So in the metaphase, the complete condensation of chromosome will be there. Morphology of chromosome is studied here, the size, shape, number, everything. So spindle fiber will attach with kinetochore. Chromosomes, they lie at the center, they lie at the equator. Now in the anaphase, because your chromosomes are arranged like this at the center, at the equator. So in the anaphase, remember, splitting, right, there will be the splitting of centromere. The sister chromatids will separate. This is the speciality of your anaphase of mitosis. The chromosomes will move towards the opposite pole. Now because these sister chromatids are moving to the opposite pole, so now they will start behaving like what? They will start behaving like the chromosomes. Keep it in your mind. Now in telophase, which is the reverse of your prophase, what's going to happen, my dear people? Endoplasmic reticulum, Golgi apparatus, your nucleolus, nuclear membrane, they all will reform. It is the final stage of mitosis. Chromosome will reach to their respective poles and they will decondense and they'll lose their identity. So it is just the reversal of prophase. Then cytokinesis, the division of cytoplasm. So in animal cell, the cleavage furrow will form, and when you talk about the cleavage furrow, right, it is with the help of actin filament, that is the cytoskeletal element. And always remember, it is from periphery towards the center. So we will use the word centripetal here. Cell plate formation is there in plant cell. Golgi apparatus will help in that. And here it is centrifugal arrangement. That's what you have to keep in your mind. This is all about the mitosis, where after the division, the daughter cells will have same number of chromosomes just like the parental cell. Now let's move to the next part, that is the meiosis, which is the reductional division. Okay, which is the reductional division. So when it comes to the meiosis, right, here it will be, remember, 2 rounds. There will be 2 rounds of nuclear division and cytoplasmic division, but just one round just one round of DNA replication. That's what we have to keep in our mind. So after interphase, there will be meiosis 1, which will have prophase 1. And when you talk about the prophase 1, so the trick here is LZPDD. Little Zara, please do not dance. So leptotene, zygotene, pachytene, diplotin, dikinesis. Leptotene, zygotene, pachytene, diplotin, dikinesis. That's something you have to keep in your mind. So ultimately, it is the prophase 1. That is for sure. The condensation of chromatin will be there. Organelles, they will disappear. The nuclear membrane will disintegrate. That is for sure. But here, because it is the meiosis 1, it is the reductional division. Actually, the point here is this is the first nuclear and cytoplasmic division, which will give you 2 daughter cells having haploid number of chromosome. Then there will be meiosis 2, which will give you 4 daughter cells. Right, so meiosis 1, meiosis 2 is there. So after meiosis 1, chromosome number has to reduce, right? So in the prophase 1, we will see that type of substages, and one question will for sure come from this part. So in leptotene, chromosome, they will become visible under the light microscope. The compaction of chromosome will continue, right? Then in the zygotene, there is the pairing of homologous chromosomes. So we will say synapses or syndesis or zipping will be there. So how that homologous chromosomes, right? Homologous chromosome having same shape, having same size, same centromere position, same genes, but yeah, allele can vary. How will they pair? With the formation of synaptonemal complex. That's what you have to keep in your mind. So here the bivalent will form. We also call it as tetrad. Why is it so? Because these are 4 sister chromatids that will appear here. So bivalent and tetrad is same, right? So here it is the synapses. So paired chromosome is bivalent. Synaptonemal complex will form in zygotene. So pachytene, which is longest substage in comparison to leptotene and zygotene, here the recombination, also known as crossing over, will occur, and that is an enzyme mediated process. So recombinase enzyme is required. So crossing over will always occur in between non-sister chromatids of the homologous pair of chromosome. Focus on the language here. So these are the non-sister chromatid of homologous pair of chromosome. So the crossing over will occur in them. So before crossing over, the point where the crossing over has to occur, the recombination nodule will be there. So recombination nodule marks the area where the crossing over will occur after the exchange of segment. After the exchange of segment, because they will not get separated immediately, right? These segments, they will not get separated immediately. Like for an example, let's suppose if this is the homologous pair of chromosome, right? So of course here the exchange of segment will occur, but even after the crossing over, they'll stay like that. So we call it as X-shaped chiasmata. What are we going to call it, students? We will call it as X-shaped chiasmata. So it marks the site at which crossing over has been done. So recombination nodule, the area where crossing over will occur, but here it marks the site where the crossing over has been done. So that's something you need to keep in your mind. This is for the pachytene. Crossing over will occur. Then diplotin, dissolution of synaptonemal complex. Synaptonemal complex will dissolve. So these chromosomes, this bivalent, they'll try to get separated, but still they will not, because still that X-shaped chiasmata is there. So homologous chromosomes separate from each other. X-shaped structure called chiasmata are visible. It is the longest phase of meiosis, which can last for month or year in some vertebrates, like even in humans, right? In oogenesis, right, you know that the meiosis 1 will start, but there will be an arrest at diplotin stage, that is known as dictate arrest. After it, there will be dikinesis. So here the terminalization of chiasmata, that is the particular feature here. It is the final stage of prophase 1, and it marks the transition to metaphase. It marks the transition to metaphase. That's what we have to keep in our mind, that it marks the transition to what? It marks the transition to metaphase. So chromosomes are fully condensed by this stage. Spindle is assembled to prepare the homologous chromosome for separation. Nucleolus, nucleolar envelope, they just disappear. Nucleolus will disappear, nuclear envelope will break. Now let's move to the metaphase 1. Remember, the bivalent will separate. So here in metaphase 1 of meiosis 1, no splitting of centromere, right? There will be no splitting of centromere and no chromatid separation. Are you getting my point? No chromatid separation and no splitting of centromere will be there. It is just the bivalent that will get separated. So in metaphase 1, whenever you will see the homologous chromosomes are attached here because terminalization of chiasmata is there. So still they will not get separated. So in metaphase 1, you will always see that there will be two metaphasic plates. So of course, after anaphase 1, it is the bivalent that has to separate. Okay, it is the bivalent that has to separate, not the splitting of centromere will occur. And after that, of course, we have the telophase 1, where the nuclear membrane and nucleolus will reappear, chromosome will decondense, but not at extremely extended stage, right? Because after telophase 1, they need to enter in prophase 2, that is why. So cytoplasmic division will start here. Okay, now meiosis 2 is the next thing which is considered as the equivalent to mitosis. Why? Because see, in meiosis 1, you know that the chromosome number will reduce to half. But remember one thing here, right? The DNA content will be 4C. After meiosis 1, in these two daughter cells, the DNA content is 2C. And then there is the requirement of meiosis 2, because we have to reduce this DNA content as well. So after meiosis 2, actually that sister chromatids will separate. So each daughter cell will have n number of chromosome and the DNA amount will be 1/4. So here it is 4C, here it will be C. So if it started from 200, here it will be 100, here it will be 50. That is what we have to remember, right? Why? Because when there is the gamete fusion, so sperm N, ovum N, they will fuse. You'll get 2N zygote. So C and C, zygote will get 2C, and that's what we want. This is the initial amount of DNA, but after S phase, it has become 4C. So because it's this cell, it is after S phase, this cell is undergoing the division. That's why we are saying here it is 4C. Okay, so mitosis, in the meiosis 2 also, the daughter cells will have same number of chromosomes just like parental cell. That is why we say that it is somewhere like the mitosis only. So in prophase 2, again, the nuclear membrane will disappear, chromosome again become compact. In metaphase 2, just like mitosis, chromosome will align at equator, right? And here, all the, right, just like mitosis, yes, here also they will align. They'll align at the equator. And anaphase 2, just like mitosis, splitting of centromere and the chromatids will separate. They'll move to the opposite pole. They will move to the opposite pole. And then there is the telophase 2, which, right, where the chromosomes once again, right, they will just lose their identity. The nuclear envelope will form, and after that we will get the daughter cells. So that is something you need to keep in your mind. This is all about the topic. So remember one thing. Firstly, there will be interphase, which will have G1, S and G2. Then there is meiosis 1 having prophase 1, metaphase 1, anaphase 1, telophase 1, and we know it very well, prophase 1 will have L, Z, P, D and D. So after this telophase 1, prophase 2 is there. So there is a gap in between, but that gap is known as interkinesis, my dear students. It is not. This gap is interkinesis. It is not the phase where the DNA replication will occur. DNA replication will occur here only. So that's why there will be 2 rounds of nuclear division. There will be 2 rounds of cytoplasmic division, but just 1 round of DNA replication. That's what we have to keep in our mind. This is all about the topic. So if you have studied it before, you can very easily revise it. You can revise it very, very, very easily. It is as simple as that, but it will give you how many marks? It is going to give you 16 to 24 marks. So stay tuned to our channel for more such videos, and whatever topic you want me to teach next, do let me know in the comment section.